Ladies and gentlemen, welcome to the NTU Royal Palm Lecture Series. Before the lecture begins, let's first welcome the host of today's event, Dr. Wen-Zhang Chen, the President of National Taiwan University, to deliver his opening remarks. 各位先生、女士，欢迎莅临台大椰林讲座。演讲开始之前，让我们掌声欢迎国立台湾大学陈文章校长，也是本场演讲的主持人，为我们发表开幕致辞。
President Nagata, President Yang, and also our Vice President, and our faculty and the student. Good afternoon. Yeah, it's my great honor to introduce our speaker for the Royal Palm Lecture Series, President Nagata. For today's lectures, we create the Royal Palm Lectures since last September, and as you remember, the first one is Nobel laureate. Ameno Sensei, okay. And after that, we did have several lectures, and every lecture I think we enjoy very much. Let me give you a very brief introduction on President Nagata. Nagata Sensei, he received his PhD from pharmaceutical science from University of Tokyo in 1981. And after that, he did spend sometimes in the United States, including the Albert Einstein College of Medicine. I think, well, recently, a lot of people, heard about that news. A very rich lady, she donated about $1 billion to that college. So everyone do not pay the tuition, and we hope University of Tsukuba and the NTU can receive a similar kind of a donation. I think the President Nagata, he brings the fortune. And after his, the post-doc periods in the United States, he returned to Japan in the National Institute of Genetics as an associate professor. And he moved to Tokyo Institute of Technology for about 10 years until 2001. And after that, he became a full professor in University of Tsukuba. And in 2013, he became the president. Up to now, it's more than 10 years already. It's amazing. For me, it's impossible to be a president for 10 years. 4 years is enough. It's a very, that's a difficult job. And also, in 2019, he became the president of the Japan Association of National Universities. It means a leader for the national university in Japan. And not only his great achievement in administration work, he also did very well in his academia work. His expertise is on the biology and the structural biology, and also the biochemistry. And he's received a very big honor from the Japan Society on that. And also he received the medal from different countries, honorary degree from Malaysia and others. And besides that, he also received the medal from our Ministry of Education of Taiwan. And I was very impressed about about the President Nagata. His not only achievement, and he's very energetic. As I just heard from President Yang told me, he fly to Taiwan, Taipei in the morning, and then come back, to Japan, and back and forth, and get with 1 day, and also he get a very big rank in 1 day. I'm jealous on that. I hope I can do the same thing for NTU. But anyway, I think that he is a very good friend of NTU, and we not only for the project collaboration. As you know, we have 3 university joint master degree program with University of Tsukuba and Bordeaux and NTU, and our director is there. Not only that, we have project collaboration, the university level bilateral meetings, and we hope we can have the joint research center very soon. I think that I want welcome President Nakata to visit Taiwan, many times in NGU, and I believe we will enjoy his lecture very much. So thank you very much. We hope that President Nakata can enjoy your day in NGU too. Thank you.
Thank you, Dr. Chen, for your inspiring remarks. 谢谢陈校长的致辞。Now, ladies and gentlemen, please join me in welcoming our speaker, Dr. Kyosuke Nakata.
All right. Thank you very much for very, very kind introduction to me. And President Chen and President Yen, these are two of our best friends. And also I have looking at many my friends and you here. And I'm very proud and I'm very honored to be here and to give you that lecture. And the Chen, however, asked me that I have to give you lecture twice. One is today about science. The other one is, he said that there's something about university policy or something, so that I will come back sometime. Okay? But today, I like that they're joking every day. But today, I'm scientist, so very serious, okay? And okay, this is my CV. Already, Professor Chen explained, and so that I skip this. And this is my special laboratory right now. And usually, president of University of Tsukuba has to leave their laboratory. But this kindly, the medical department gave me the chance to continue science. And last year, we published that very nice paper for the year. Probably you don't know, and science-related article and EMBO journal articles. That's probably my last one. I couldn't do that science no more. And I don't know that who is the audience for this lecture. I imagine that probably young guys and staff members. That I started with my career. How or why I studied in virus and what I find out. And this is anyway, this guy is my teacher, Professor Masaatsu Yamada, who had been working in Colorado for a long time and coming back with their HeLa cells. This is the first time, Japanese use HeLa cells, which actually this guy taken. And this is myself. Okay. And when fourth year of undergraduate, I worked with the Masaatsu Yamada. And my project was how a cancer cell merge. Very big project. And I didn't know that, so that we try. Anyway, we try to get at the piece of data. And this is a famous slide. Usually, normal cell grow, and sometimes stop, then going to death. However, sometimes you have some mutant cells, cancer cells, which can get that immortalized long life. And so something is happening. And we want to know. But why this is possible? Then Yamada sensei asked for an undergraduate student, which do you like? So that I like this one. DNA synthesis phase, how DNA or synthesis is initiated. And these guys take some parts. And probably you know that some guy. This is Ono, used to be the professor in the Kobe University. He is a guy first clone the PKC. And also this guy is the former Kirin Kyowa CEO. And these my friends, actually, same grade. And then as everybody take each phases. And I took that the S phase. And I have been working for the 1 year undergraduate, 2 year master's, and 3 PhD course. But I have only this. This is my only data. Yeah, because here, what I want to know is what is the switch for DNA replication initiation, and nobody knows. And even we didn't have any sequencing technique. We didn't have anything molecular biological technique. So that's I think something there. How I prove that. And left slide. I treated the cell with the protein synthesis inhibitors. It had been reported that the protein synthesis is essential for DNA replication. So something is newly synthesized. So that the cell now decreases the replication activity along with the duration treated with the protein inhibitor. Then take nucleus here, and then we put something, and if DNA replication starts, something there. In fact, I take some nuclei here, and I put extract from S-phase cells, and then DNA synthesis starts. So showing this, probably something there, but I couldn't know what is it. Okay. So as I said, that there must be a factor for DNA replication initiation. Okay? Then I have been thinking. I didn't have nothing, no technique, no molecular biology technique, and something else. So that I saw that probably virus is a probably good tool instead of the cell itself. But because it's small, so maybe I can do something. And as you know that the virus itself cannot grow. Virus has to infect cell, which already means that something is essential for the virus replication. Okay? This is a very simple idea. And so not only virus factor, but also the factors from host cells are required for virus replication. It means that some virus factor probably interact with the host factor, then functional. And this situation is true for every step of the replication of the virus. For enter, probably we need, actually virus, not virus, but virus needs some help of the cell and replication or something. So that I decided, supposed to be the using virus, I can know that a factor which is essential for DNA replication. And by the way, this slide summarizes how many Nobel laureates from viral research. And so many, actually. Probably most famous one is split-in, as a splicing mechanisms found by Sharp and Roberts by using adenovirus. And this is a bachelor degree here myself. And I got the master degree here and the PhD degree here. So already some virus contribute life sciences. Then after still many discovery from virus research. So true, probably virus is a very good tool to know. Then I choose the adenovirus at the time in Albert Einstein College of Medicine. Then I skip everything. But this is the final slide after 5 years working in Albert Einstein College of Medicine. I completely dissect of the adenovirus DNA replication system, and I know that the 3 viral factors are involved. In addition, I found that there are 2 kinds of the host factor, which are essential for DNA replication initiation of adenovirus. And first one is I designate nuclear factor 1, NF1. Second one is NF2. Then this is actually, I loved data. And this is a footprinting experiment. And I skipped the detailed explanation, but as you can see that something is happening. DNA is cutting the DNA, but in the presence of the protein, some area is protected from the DNA's digestion, it means that protein bind to the a specific area here, the other strand here, and these are samely digested by DNA. So it's binding DNA sequence specific. This is the first case of the DNA sequence specific binding protein. NF1 is later is identical, shown to identical with the one of the cat binding transcription factor. So adenovirus use this NF1, so cat box binding protein, as initiation factor for DNA replication. And this factor specifies the origin of DNA replication. With this study, I realized that, oh, yes, we can manipulate the entity of the virus because it's small. And also, we can reveal that real involvement of the host factor in the virus. This is the first case. And so that we may approach novel function of even the cellular factor through virus research. Already, you know that a number of the finding was awarded by the Nobel laureate. And so that since then, I took that the number of the virus is a tool to know the host function. Adenovirus, human papillomavirus, cytomegalovirus, influenza virus, mumps virus, measles virus, and so on. And I want to know still what is the DNA replication switch. And I already know that the NF1 is the sequence-specific binding protein, and also physiological role in cell is probably transcription factor. And at that time, I realized that something I make a mistake, because I set up that the DNA replication system is adenovirus DNA itself. But adenovirus DNA is naked. No, never naked. Always forming the chromatin structure inside the cell, so that maybe I missed something. The same thing true, genome is not naked. Always compressed with the histones or other nuclear protein. So therefore, real template is not DNA, but chromatin. So that I changed my template from the naked DNA to the DNA protein complex. And this replication transcription system for naked DNA never worked. So something missing. So I try again, purify of the protein, which a remodel of the DNA, partially at least as is naked DNA. And then this protein can stimulate replication and transcription. And this protein is a template activating protein factor 1. And later I can tell you about this is the histone chaperone for H1. And sometimes H3 and H4. And the physiological role in the cell is epigenesis, epigenetic control. Then this is the amino acid sequence of template activating factor 1. The red letter shows that the acidic amino acid. Here is the long stretch of the acidic region. So DNA binds to the basic protein, such as the histone. So this acidic protein is bind to the histone, maybe, so reveal from the DNA. It could be. So that I test that. This is a model. So I looked at by the AFM, and it is true. This is the adenovirus DNA protein complex, and treated with this protein in vitro, and now you have the relaxed form of DNA. So this protein remodel of the chromatin structure in this type of relaxed form. Now, replication and transcription factor can access. Then this reaction is going. Later, this protein shown to be very important in a variety kind of chromatin reactions. And not only adenovirus, but also the herpes virus chromatin remodeling use this protein. And also this protein is plays a lot of roles in a different kind of the chromatin reactions. I skipped this, but this can be already published in many papers. And then after, I purified another temperature activating factor. And temperature activating factor 1 and 2 and 3. And the 2 is fortunately identical with the NAP1 nucleosome assembly protein. And the TAF3 is exactly the same of the nucleolar protein. And still we don't know exact function of this TAF3. But the location is different. And this column just summarizes that location and a function in adenovirus replication, and histone binding, and histone transfer or not. And everything, these guys are histone chaperones. And other later studies show that histone assembly and the assembly from the chromatin is through two pathways. Replication dependent pathway and replication independent pathway. And some factors are involved in replication dependent histone replacement. And sometimes these factors are involved in replication independent histone assembly or the assembly. And therefore, we found that the very nice players. And just I only focus on the TAF1 as the histone H1 chaperone, because still we don't know completely what the protein is the real histone H1 chaperone. And at least this one is a part of it. But still 2-3 other histone chaperone candidates are reported. And this summarizes that what kind of the histones inside the cell, germline, somatic cells, and aging cells. And this is a nucleosome structure, and histone is binding to the linker between two nucleosomes. Okay, so that histone H1 is the compact of the nucleosome structure, or relaxed of this structure. And this very simple experiment. So which chaperone binds to the H1? And the answer is very simple. And H1 frag tag pull-down experiment show that TAF1. So NAP1 doesn't bind, or CAF1 doesn't bind, ASF1 doesn't bind, but now TAF1. So that the TAF1 is the very strong candidate for the histone H1 chaperone. And just show you a very small data. This nucleosome, these are nucleosome, is bound with the TAF1 to move H1 in the presence of the TAF1, move to the nucleosome H1 complex. Okay. And more optical observation could be give you. And this is the bleaching experiment. The protein is labeled with the, for example, GFP, and then bleached with the laser. Then after, how this protein is reassembled in the same location in the cell. And probably this is more easier to see. If you look at the first one, usually this one is the H1 EGFP labeled cell. And if laser show a bleach of this portion, now bleached, now recovering this way. However, if you decrease the amount of TAF1 from the cell, now bleached, but not recovered quickly, still not bleached. And NAP1 is very quickly, so that this kind of the curve. So usually histone H1 is recovering with this dynamics. However, in the absence of the TAF1, histone H1 is more slowly recovered. So therefore, again, show you that the data, piece of the evidence that TAF1 is the histone chaperone. And this is the static, so no problem. And overexpression of the TAF1 is increase the rate of the moving of H1 inside of the cells. And so that TAF1 is involved in the histone H1 assembly or detaching. Well, still we don't know that the other protein is or are involved in. And in this section, the final slide give you that the more interesting data. TAF1 relaxes at the chromatin structure, but also TAF1 relaxes of the sperm condensed chromatin like this. Usually, this happening inside of the oocyte. Sperm is coming into the oocyte, then they condensed, then making the chromatin structure and paired with the oocyte chromatin, then start dividing. So therefore, TAF1 could be remodeling of the condensed sperm chromatin. And this one is the data, a piece of data about TAF1 knockout mouse. This is a male case, so that once the bone after at the embryo days 12.5, completely the difference between wild type and knockout. This is hematopoietic cells, but not really so many hematopoietic cells. So maybe involved in the differentiation of hematopoietic cell. But these are ongoing. And still, I have a question. What is the function of NF3, which is located in nucleoli, nucleus? So that there's something there. And probably this is involved in the assembly or disassembly of the nucleolus, but which is ongoing project. Okay, and I am the child of the central dogma, and in 1962 this central dogma was proposed. Then after people engaged how DNA replication is initiated or regulated, how transcription is regulated, how protein synthesis from the messenger RNA. And still some questions there. But I'm really interested in, first time of this session. Later, I'm interested in this part or something like that. However, this central dogma does not describe another feature. That is, Dr. Bolchimor found that the reverse transcriptase, so that the central dogma is now add that the different reaction RNA to DNA. And also, Dr. Haruna found that the RNA is replicated from RNA, so RNA replication is also true. So that now I was interested in, well, I know some about DNA replication. So I changed that now, RNA replication, how the RNA is replicated. And so I choose that again, virus, so that the influenza virus could be very nice. And this is a schematic structure of influenza virus genomes, 8-segmented RNA genomes. And more importantly, this RNA is template polarity. This is not coding strand. It is the template for coding the sequence. Therefore, influenza virus should have RNA-dependent RNA polymerase itself with genome when get into the cells. Otherwise, cannot replicate because this is just a template, okay? Negative strand. So I'm very interested in that. This is the cartoon of the RNA genome structure. RNA is not naked, complex with the 4 proteins. 3 RNA-dependent RNA polymerase proteins and nucleocapsid proteins, and this type of the structure. And so negative strand RNA and 3 polymerase protein, 1 nucleocapsid protein. These are components of RNA protein complex for replication transcription. And we purify of the RNA polymerase. Then characterize of the function of each subunit. This one is already known that the PB2 is involved in the CAP1 structure snatching mechanisms and the PI, which is involved in the replication. The PB1 is a catalytic subunit. And this is the mechanisms of replication transcription. For transcription, as I say that viral RNA is negative strand, so template. Therefore, polymerase has to lead of the RNA along with the RNA template, making messenger RNA. And the replication process is different. The vRNA, viral RNA, is once copied into the complementary RNA. Then after, again, copy of the virus RNA from the cRNA. So this is the replication process. And I set up that the fundamental system to find out of the factors. With the RNP complex, polymerase and NP and RNA can catalyze the transcription but not the replication. So we miss something. However, if you mix this complex together with the uninfected nuclear extract, now you can resume of the transcription, so higher than this alone, and replication occurred. So again, replication factor in uninfected cells that we have. We help the influenza virus replication. Amazing. And I tried again to find out of what factor is involved in the initiation of RNA replication. And so that this is an assay system. In absence of their nuclear factors from uninfected cells, viral RNP do nothing. As a function of increasing amount of the host factor, replication occurs. Actually, this is a purification scheme. And now you have the 8 segment replicated. Then identity of this factor is MCM complex. The MCM complex is the complex for S-phase initiation regulator. Again, the DNA replication regulator, but this one also involved in the RNA replication of influenza virus at the first stage. So vRNA to cRNA. And if you knock out of the MCM, now viral or cRNA synthesis is decreased, while vRNA synthesis, vRNA messenger transcription system is not affected. So that exactly this, in this case, just knock out of the MCM3, but probably MCM complex is involved. And then after, I tried to identify that the factor for second step, cRNA, complementary RNA to the progeny vRNA synthesis. Again, I purify like this. And then finally, I got the factor. And then this factor is already isolated as PP32 or other names. And the structure is very similar to the TAF1 that's involved in the adenovirus DNA replication, body and acidic tail. Body and acidic tail. And I suppose that this acidic region is quite important. Then this slide is, I like this slide too, because the cRNA template, if you use that the cRNA as a template, so some vRNA synthesis is observed. However, if you use that the vRNA as a template, you can't. So it means that this factor is specific for second reaction of cRNA to vRNA. And terminal is the, actually I skipped at the detail explanation, but the terminal of the product is PPPA. So start with the ATP. Okay? Then, these guys, the replication factors. And then I try find out of the factor for NP. And NP bind to the RNA cooperatively. So this process is very slow. So therefore, some facilitate of this complex formation. Then I purify again using the transcription stimulation assay. And then I purify finally two proteins. One is the P48, the other one is P36. And these very pure protein. Then, and this P48 is exactly same as UAP, which is involved in the splicing process, the RNA splicing process. And the binding phase is same. NP has this primary amino acid sequences, but if you make that the alpha helix, MTGY is the same side. And this factor is UMP is P48 is also known as the UMP, which is binding to this splicing factor, and this surface is also forming MTGY. So this is the binding surface of this protein. And I skip this one. So that again, this P48 protein is the chaperone for NP to bind to the viral RNA. And the P36 was later shown to be involved in sequence recognizer. So that this P36 recognized the viral subsequence, then transfer this 48 to the viral RNA to make that the RNP complexes. And however, another question is, during the replication, RNA has to be encapsulated. So then going to the progeny variant. So therefore, something is happening during the replication. However, in absence of just adding of the NP to this reaction, nothing is happening. Also, the NP does not bind to this nascent RNA structure. So probably this is the we found that yeah, and this is a piece of data in the presence of the UMP56, which is a P48. Now you have the long strand nascent elongated product. Okay, so by cartoon something like this. In the absence of this chaperone, NP cannot bind to the nascent RNA strand. However, in the presence of the P48 or UMP56, NP covering of the RNA and making this capsid structure. So that this is sort of the elongation factor. That's why I picked up this protein as the transcription elongation factor. And this one is same thing, NP chaperone, but the system is very interesting. I switch the yeast system. Okay, in this slide, I reconstitute of influenza virus genome replication transcription inside of yeast. Question whether yeast can help or not. Yeah, it is. So that yeast help that the replication and transcription of influenza virus genome. So it means very good because the yeast is a very good genetic tool. So therefore, I tested this replication transcription assay using single mutants of the yeast. Then we found two mutants cannot support influenza virus replication transcription. Cas2, which is the UMP and LAF2, exactly the same homolog, of course, so that we picked up. And again, another one, PRP18, which is also very important by genetic screening using yeast. And this protein is that, this is just a cartoon summary. This protein is chaperone for NP, but not nascent RNA. This does bind to the NP, then organize of the cooperative binding of NP. So this is a replication independent NP binding factor. This is a summary what I have been doing for the replication, for the transcription. For the transcription, we have found that here, I skipped this one, but factors. And also for replication, first stage and second stage, we found that here factors from host cells. Okay? And so this is a summary what I have been found. So these factors from cell for adenovirus replication. These are cellular factor for influenza virus replication and transcription. And then after, finally, I have to contribute for the medical sciences. Finally, that was just my science, interest. But now, so because here we determine the 3D structure of the part of RNA polymerase. 3 RNA polymerase complex is functional, and then we determine the PA and PB1 contact site of the 3D structure. And this PB1 is binding into the PA hole from the N-terminal domain. This is actually the first case of the 3D structure determination of RNA-dependent RNA polymerase. All right, this is nice for the drug design. If you can that the design of the drug binding into this hole, maybe this polymerase cannot be reconstructed, means the replication is inhibited. And this is a different view of the this structure. And also we determine that the PB1, PB2 contact site too. Let's skip that. And this structure is reveal that the contact amino acid, so that we mutated of this amino acid which is involved in the contact between these subunits, and exactly knock down reveal of the replication activity. And so now I contribute to the medical sciences. And for control of the influenza virus, we have two strategies: vaccine and anti-influenza virus drug. The both has the merits and the problems, but most difficult to be solved. The problem is always the virus, drug-resistant virus, emerged very quickly. So that we need the design, the antivirus drug, which cannot be escaped by virus. How? And then this slide tells you very important information. This again shows you that the blue parts, these blue amino acids are involved in the protein-protein interaction, forming the red one is the mutation between 1934 strain and the 2003 strain, so 70 years, over 70 years. And there is many mutation sites. However, you can find any red and blue merged amino acid, suggesting so blue one cannot be mutated. Maybe structural reason. Well, you have many, mutations. However, this blue amino acid cannot be mutated over 70 years, which means that these cannot be changed. Functionally, very important. So, it means that, again, if you can find out of these chemicals, which is binding to instead of the N-terminal domain, the PB1, this is very nice candidate for anti-drug. Then, in silico screening, more than 500,000 chemicals in silico, we found that there are several candidates. And then we test the antivirus effect. And this is a plaque assay. In the presence of the chemical C, you don't have that no plaque. Means that stop of the influenza virus replication. And this data is now shared with the pharmaceutical company to develop of the new generation of anti-influenza virus drug. Okay, almost same, last. And another strategy is right now, I'm thinking is same thing true for the host factor and viral factor contact site. Because the host factor is encoded by ourselves, which cannot be mutated. And so that it means that this contact site of the polymerase is also can be conserved. So therefore, maybe this is another target for designing the new novel drug. Okay? And I would like to skip that because of the time. And I am satisfied about control of replication and transcription, but not still satisfied. Because the very big question here, I skip that. I told you that the influenza virus has 8-segmented RNA, which has to be assembled as the one set into the progeny billion. Nobody knows that how these different 8-segment package into 1 billion. If you inhibit of this process, this can be the very good target for the antivirus drug. So therefore, I'm very interested in right now how a set of 8 segments assembled. We don't know. Nobody knows. So that we tried, discuss, or and so that I did something, but I skipped that. Finally, I just emphasize again, using virus. I believe that probably virus is a good tool to know of the cellular function. And that was probably true, as I show you that number of the evidence that I gave you. And also, this extend into the viral control era. As I told you that probably some specific function is connected to the some specific inhibitory mechanism, including the drug or something like that. Okay, so that I'm still interested in how a set of the 8 segments are assembled. And lastly, and this is the final slide. Right in the University of Tsukuba, some member group focusing on the future prediction of virus evolution. How the virus is changing in the future based on structural feasibility. And amino acid or DNA sequence, RNA sequence can be changed very easily. However, functional change cannot be accepted by virus growth. So that's based on computer science, what the change can be acceptable for virus. And then that has to be tested. So that in the presence of the some specific, for example, antibody, so escape mutant is March. But we make that another antibody against this mutated virus, then what kind of the mutated virus merged and repeated? And then now, based on the structural bias, maybe we can predict next mutation of this coronavirus. So this is my, right now, this is my actually job with my research groups. Okay, so thank you very much.